Okay, I would say it's 1:05. Just let's start, and then I think we catch up wise. Go over. First of all, we started last week introducing first examples using using notebooks, and I put this demo notebook on the website, and just want to repeat some of the things because some things I mentioned, but maybe they have been very new for you, and some things are new, and this might confuse you. So let me just go briefly over this before I then actually start with the actual lecture of today. So that means this here is a text cell. So currently I executed all the cells, so it's rendered. If you click it, so you can see it. It's a simple markup, or actually markdown syntax to create text things, and so you can integrate all kind of texts, even tables, very nicely with other things. Okay. The only thing is that it then always creates a code cell below, but I think so. So what is shown here actually, if you want to do or have to do calculations, these are things you use very often. So the NumPy package, which provides a very nice C-like array, so you can create array numbers, and then it also has a wealth of mathematical functions. So here actually, this is also a very useful thing, because you can create handy aliases for lengthy package names. So that means the original name is NumPy, but we want to access it under the alias NP, so less work for you to type. This here actually enables floating point division. Otherwise, you might have an integer division, and this can lead to unexpected results. So who needs the result of 3 divided by 2 in integers? So 3 divided by 2 as an integer division gives 1, and this is of course not the result you expect. So this should be used quite often. So, and then actually this is only that's to demonstrate some fancy aspects. That's to import some tools necessary for functional programming. So here it's very simple. I create 10 random numbers. So that's the function rand_n gives you a standard, normal, distributed number. That's actually the whole thing is to generate some toy data, and here I use this simple syntax just to output the two variables so that you can see what's going on. Of course, if it's a real project or if it's in the code, you don't need this, but that's only for make it easy to understand. So then here again I use this list comprehension syntax. So I want to create, so the idea behind this, I create some more or less random numbers for, or slightly correlated numbers for body height and body weight. So that means to this base height of 170 centimeters, I just add a random fraction. So this is created by iterating through these random numbers, then times 10, and add it to the thing. So I get a height vector, and here I show the height vector, and the same for the weight. Then you can also even make it a little bit more complicated. So here to create the BMI, it's abbreviation for body mass index. Here we see already one function imported from this func tools, the zip function. So there is an important change between versions 2 and 3. So actually the idea is, for example, if you have two separate lists, but they somehow correspond to each other, that means you want to apply maybe the same function on item number 1 from one list, item number 2 from the other list. So this zip function combines them and returns you the pair in the right order, and you don't have to write a separate code or create another data structure with a list of lists. So yeah, here in Python 3, this has become more or less lazy, so that this returns you actually an iterator. So if you iterate like here, so you create here a pair, and here immediately you split this pair in two variables a and b, and can process this. This is exhausted after you iterated once. In Python 2, it creates actually a new data set of pairs, but this is here more or less the volatile, so whenever you want to use something like this, you have to redo this, or I show you later down how to make it permanent. So this is then the body mass index, okay, create some kind of patient's ID, so simply I count up 1 to 10. Here you see the syntax how to make it nice, so sometimes you prefer formatted output. For this, of course, you can write your own function. Means first counting the number of digits of this, and then adding the right number of padding characters. But you can also use this. So that means here is the format string. So it means here it's fill up with zeros, and it's actually you're talking about a 4-digit integer. And then this is a little bit maybe unusual, but it's the way it does. And then you provide the string you want to format in this format function. Then this gives you here nicely formatted output. Okay. So then the second useful function imported from the functools package, it's the map function. Actually, if you have a suspicion that this has to do with map and reduce, you are right. This is the map function takes one function and applies it to a list. So that means every element in this list is applied to this and returns a new list. So again, you save time to write a for or a while loop. Okay. And then I want to create some output data, and here you see the syntax. If you want to, you can, for example, in one step combine 4 different lists into a kind of array here. But to make this permanent, means that you can reuse it several times. You have to immediately then convert it into a list. So actually, this here is now the list of lists. If you omit list, this gives you some unexpected behavior, so that means this would still work, but then afterwards this is empty, our data is empty because this iterator is exhausted, and you cannot write it to a file anymore. So that's actually was just the prelude to create some kind of CSV-like data. So sorry. Okay, so then another useful package is this pandas, which provides a spreadsheet like functions. That's actually the whole thing to read in the spreadsheet. Then first, since it has an internal rich structure, it's not exactly in the way you want it, so that's you can simply output it. So it has already nice formatting, but it introduces an own index. If you want to use these indexes, we just do some kind of data manipulation. So that means we say, okay, that this index should be the values from this column. So the columns can be addressed using the square brackets. So that means we copy this data into this index column. Okay. Also keep the name, so you can have a separate name for the index column. And then here we just say, okay, we don't want a name. This removes this first name for the index. And then, of course, we have this now twice. We simply, again, delete this whole column and have nicely shaped our data. Okay, and then we can subject this very easy to all kind of things. So typically you use pipelet from the matplotlib package. So again here save time with a shorter prefix. If you're unsure what you actually currently have, because you don't see it immediately on the identifier, you can always use the type command to find out, okay, you're actually working with a data frame. The selection, row selection needs this log function. So you give then maybe a key value, and it selects you the whole line for this, and then you see already you have a nicely named and structured output. Okay. Data types. You can also maybe, if it's a very long table, and you only maybe want to have the head. So you can also adjust the number of the things or the tail, and you can also retrieve the index if you want it. And so this is already a quite nice function, because you can apply or calculate a lot of statistics about your data set without too much programming. Means that how many items in each column, the mean of each column, standard deviation, minimum, maximum, and here the quartiles. Okay, transposition is also very easy. Okay, that's how you do this on a single column, or actually on the whole columns, only the mean. So there is a lot of other functions that it's only to demonstrate this. So now that's also quite typical to, if you want to display something, you have to create corresponding pairs of x and y values, and of course, you have a.
Where do we find all these packages and methods, functions? Is there something like
That's part of the standard. If you simply, for example, install this Anaconda, that's all already pre-installed.
Like an API where you can just have the index to go through all the packages and sort out what you need. I mean, if I don't know if this exists, so where can I look?
So I think these are standard packages. If you don't handcraft a Python installation on your own, but take one of the common ones.
Can I find the list of packages, the list of functions, methods?
Oh.
Is there a button for it? Is there
No, not here. You can.
I just need something like to look at, to choose if I can make use of it or not.
I think there is a website called Python Index out there, which lists all the available packages on your installation. You can go to the directory structure and see what modules are listed there. Then you can, for example, that's actually not intended for this. So this is currently just a running Python interpreter on the shell. So for example, if you say import, then say plotlib.
Therefore, already I need to know what exists to import.
Yeah.
That's what I'm in trouble with. That I just
okay, matplotlib. Yeah, mat. The thing is, if you work over time, you know the typical packages. You search if you are needed for a certain function, search the web, and then.
There's nothing like the Java API where you have all the listed.
Yes, you can also consult the Python API reference, but this is, of course, you need maybe 1%, and then you get.
But there exists one.
Yes.
Okay, that's.
So and then you can do matplotlib.pyplot. So you see these are only in all the symbols, including constants, submodules, functions in the pyplot. Modules you can figure out this on your installation. So once you imported this, there are also various resources on the internet. But it's maybe harder to find something in this index because it's so big than if you have maybe a handful of modules where you know these do your work. Okay, so I want to create some files, and for the diagram here below, I simply want to have the ordered the values ordered on the x axis. So means first I combine this weight vector and the height vector into this zip object, and then I create immediately a dictionary. So the dictionary actually takes pairs and immediately takes the first one as a key and the second as a value. Then I extract only the keys, let it sort, and then if I iterate over this sorted weight list, I got here the corresponding heights. So this again, I use here this list comprehension syntax. You could also have it as a separate variable. So, and then that's a very simple, I think, procedure how to create a plot. So you call the plot command. You give the vectors for the coordinates. You can set colors, markers, how the points are marked, and list style, title, label, and then you only have to apply show, and then here you are, and you have a diagram like this. Okay, so that's weight versus height, or the other way around, it's then body weight, height versus weight, and so it's you don't need that much effort to have already a visualization. Of course, there are other diagram types like bar plots and so on, but I hope you can make use of it. Okay, so then let's go actually to one of the two main talks. So I have one talk about this sequence databases in detail in GeneBank, and then a second part will be most likely after the break about XML technology, and I think you will also see in the end how this fit together. Actually, originally I concluded this lecture with a 50 minutes webinar, but this year I try to give talks and let you the webinar do as kind of homework. I have to say immediately this is very important that you do this. We can also watch it here, but last year I had some critics that students preferred to watch it at home and would have maybe more stuff presented here. So we can have it here together, and then you can ask questions afterwards for this webinar, or we can proceed as is ongoing. So do you have an idea? Would you prefer to watch this webinar here, or would you like to watch it at home under your own control? Okay, so we have little feedback. Let's make a small vote. Who would like to see the webinar within the lecture slot? Please raise your hand. Okay.
I have a question.
Yes?
What is the content of the webinar?
The webinar is about the e-utilities provided from the NCBI website, which is a kind of API to access the NCBI databases, and this also fits together with the XML stuff and the automatic access to this. So, and I think this is a very good visualization where you can see what you can do with this, and so, and I think you could not really understand this without watching this webinar. So I would consider this mandatory. So that means you would like to see it within the lecture.
Yeah.
Okay. So okay, second one. Okay, then who would like to watch it at home? Okay, so that's the majority who wants to watch it then at home, so I will go on. Okay. First of all, about if you talk about GenBank, you should maybe start a little bit earlier and also consider the National Center for Biotechnology Information, in short NCBI. So this is actually a very huge facility which provides support for all kind of life science and biotechnology, and came up with the first idea in the middle of the 80s, and was funded as a division of the National Library of Medicine, which is also one of the national institutions in the US. Inside the National Institute of Health. National Institute of Health, I think, is the umbrella authority for all kind of research organizations in the context of health and life science research, and so also they provide a lot of the money. So it means it was started with as a clear political mission, and actually then founded in '88. So you see, first ideas middle in the 80s, and funded close to the end of the 80s. This took quite some time, and the director was appointed David Lippmann. Okay, so that's about the goal of this mission. The mission, so that means design, develop, implement and manage automated systems for the collection, storage, retrieval, analysis and dissemination of knowledge concerning human molecular biology, biochemistry and genetics. And this is actually, that's quite unique, that you have a political mission to provide service and infrastructure to the community. So typically you get funding for very specific projects, and then afterwards the project is over, and you have to look for yourself where you get funding, or how you really can provide this as a sustained service. So it also supports computer-based information systems to do research on all kinds of methods, and represent biologically important molecules and compounds. So that means there's a kind of official order to maintain databases and actually help the people working in this field, and and collaborate on the international basis. So I think there is only a second institution, the SIB, the Swiss Institute for Bioinformatics, which was funded, I don't know exactly, but it was, I think, around maybe 20 years later, which now more or less hosts the EXPACY thing. So this is the second large infrastructure unit, which is also available for people from outside. But so this is still quite unique, and the size again. So just to give you an impression about the timeline in the terms of the internet. So BLAST was developed in 1990. So, and GenBank moved to the NCBI in '92. So here the first website came, and then slowly the genomes came in, and other projects like the online Mendelian inheritance in man database. So that's a collection of severe mutations. Then later on PubMed as a literature index, and then you see much later on human genome, PubMed Central, and here this versatile access interface. And then public access genome, 1,000 genomes project. So actually, this was one of the big, preconditions to make all these other projects work. And then I think at the end of the 90s, for example, Google came up. So this is already much older. So NCBI currently hosts a vast bunch of resources. You can go to this URL and look what they actually all provide. So I think it fills nearly a whole page, and you can group it according to various criteria. So some are project-centric and contain also metadata about the projects, method-oriented, or you can also search topic-oriented. And so they have databases, downloads where I can download tools, softwares. or his own submission things, but they also have a very good instruction section with a lot of how-tos and descriptions. Okay, so back to the very beginning. Originally, it was not GeneBank was not funded at NCBI, but at Los Alamos Laboratories by Walter Goat. So actually, I spent I think more than an hour to find a picture, but this seems to be the only picture where you at least could get some of his face. So it was in the pre-digital area. So in I cannot remember when did IBM release the specification for the PC? I think it was in the beginning of the 80s. So that means they started this database where there were even no personal computers available. And then it stayed 3 years there, and then so it was the first idea, and then was released first in 1982. And with 2,000 sequences. Can you imagine what does this mean? I think 2,000 sequences is it's a tiny fraction of the space you have on your mobile. So, but at this stage, this was really an achievement, and the distribution was maybe done on magnetic tapes, or then I think floppy disks already existed at this time. So, who can remember floppy disks? Okay, so at least you still know it. Yeah, okay. So, and here I found this. So in 2002 they had this 20th anniversary, and they remembered this May 9th of 1980. Oh, actually that's nearly 37 years ago. When they celebrated the 10,000th base in the DNA sequence library. So I think for nowadays, we will see later, you can add a few more digits here to this. Okay, so this was the historical reason. Now let's look at this now and the growth. And actually, you could see already here they originally celebrated 10,000. So 10,000 is okay. It's somewhere below here. Now we have several orders of magnitude higher. So this is the data for the latest release, and from 15th April. So now we have, what are these bases actually? These are thousands, millions, billions. I think 231 billion bases in the GeneBank. No. Bases in GeneBank. Yeah, and even more. So that means, is it? Okay, maybe I have to check if I made a typo with this comma, or if I just skipped a zero here. So if it's billions or trillions of bases in the whole genome shotgun sequencing project. So actually, originally they were included, but now they are distributed separate because it would even blow up the size of GeneBank, but of course, so that's in a different section, but this also contributes to the available number of sequences. So this is the comparison. So, or was it current release? Doubling approximately every month. Oh, this is here the basis. That's the basis graphics, and here it's the sequence base thing. You see, okay, the growth of this WGS project still now has outpaced the number of the sequences in GeneBank. It looks that we run slightly into a plateau, but okay, maybe the things are transferred from this database to this database, and so we very close to the 1 billion number of sequences, and so still doubling nearly every 18 months, which is nowadays faster than the growth of the computation power. So it means somehow there is a gap opening between the available sequence information and the available of computing power. Okay, so there are several citations out there. So typically you take the latest one. So the latest one is here from 2016 officially. So then they typically update this and tell you how many sequences are in there, how many sections, and so on and so on and so on. Or if you want to cite or refer to the NCBI service in more general, here that's the different things. Actually, GeneBank does not exist on its own and in an empty world, but actually it's part of this international nucleotide sequence database collaboration, in short INSDC, together with the EMBL nucleotide sequence database, which is in Kingston. I mean, it's part of the EMBL, but this is located in Kingston, and the DNA database of Japan. So these three databases are the reference databases. So whenever you want to publish work about sequences, DNA sequences, you have to deposit your data with one of these three databases. Depends a little bit on your personal preference or maybe on local relationships, and they frequently update and synchronize their data. So it's not always exactly the same data in all of these three databases, but they typically they synchronize and they exchange data with each other. Okay, so I only added here the last numbers because actually there is no simple way to do this. You have to look to the release notes and then manually add up the data. So to see the growth from I think a year ago, or was it 2019? No. So 2013. From 2013 in hundreds to release 197 to it's I think now it's 200. Yeah. So okay, it was quite old. So within the 3 years, actually the number of nucleotides growed by the number of 4. And you also see here is a steep increase, especially also for the transcriptome shotgun data. So these are the fastest growing data, and here are the rates. So, and here are several sections from this other things. So these are not distributed with the release, but you have to retrieve it separately. This is just to give you a comparison about the top hits. So you see here, I think it's not a real surprise, the organism with the highest representation is Homo sapiens, followed by mouse, rat, and then here that's cattle, mice, pig, that's zebrafish, and then here wheat. That was released 2007, 1 year ago, and then actually that's the current release 2019. We have 2 new members on the top in the charts. So now what was a new grain species? It's actually Gerste, but I don't know the English name by now. So this joined the thing, and very surprising. I wanted to look this up, but I haven't done it yet. Maybe this new organism, which is quite surprising. If you looked at this, has only 72 sequence entries, but already that number of base pairs. Actually, here again, the whole shotgun sequencing projects are excluded. So that's rice. And here now, for now, E. coli is within the first top 20. So I think that's also a surprise, because 1 year ago we still had Arabidopsis, but no bacteria in here. So now E. coli made it into the top 20.
I think it's barley. I guess it should be barley.
Oh yeah, barley. Okay. Barley. Okay. Thanks. Okay. So as we see, the this huge database. Actually, if you download it, it's a huge set of text files or compressed text files, and this is organized in different sections. So it means it's roughly, I think now, more than 900 gigabytes. So it's divided into so-called divisions. So if you're interested in bacteria, you should only download the files from bacteria or from the BCT section, and you would have to download the 178 files. And these are here, actually, that's the recent numbers. So bacteria now grow to 350. You see, from 178 files up to 350. That's the I think the context also grow very much. This was removed from GenBank, higher throughput, and now phages gained more interest. So that means, especially from many of these environmental projects, people are realizing that viruses are very important in the ecosystem, and so that's reflected to this growing number of files. Yeah. Okay. So plant, and then there is a growth, but not that much. Nowadays we have 2,225 text files for the whole database, and that means because here I think some files were removed. Okay. So if you really want to have it on your disk, you can download it by FTP, but be careful actually, because uncompressed together it's 821 gigabytes, and that's maybe a tribute to the history when people were or had no good internet connection and sending around disks, optical disks. So they provided in chunks you could put on an optical disk. So maybe that's outdated, but it's still the way it is. So that also explains then the large number of files you have this, and then they are the size you then have to import it or to join it again. So each file has actually a header and a body. So that means the sequence entries for the division are described in the header. So that means that's the name, GenBank bacterial file number 1.sec, tells you how many sequences or actually how many loci are described. So that's here the same as the sequences, and how many bases are there in the sequence. And then the sequence entries are followed. So now I want to cover the GenBank flat file format in a little bit more detail, because that's still one of the most common used files to exchange this. So of course there are more sophisticated formats to exchange data. We will see, or we will have a short look into XML technology at the end of this lecture. But flat files are still the kind of common currency to exchange information. So actually, to understand some terms, so one sequence is typically stored in something called sequence record, or in a sequence entry. This consists of many records, and a record is simply a line in this. So each line has actually two parts, means header part or field ID, and then the content. So that means typically this is a fixed format. So the columns means the first 10 characters are used to determine the type of the field, or the type of this line, and then the remaining line is the content. Okay, so typically there are keywords and sub-keywords. A keyword, that means it's a more important identifier or line type. They start in column 1. So for example, reference is a keyword, or then you can have sub-keywords. That means if you want to specify or you can further modify a line type, and then they start in column 3 and they are just indented by 1 or 2 columns. So for example, if you describe a reference has authors and maybe also a title, then this is then sub-keywords to this. Okay. Or can also begin at column 4. Yeah. So if you have blank columns in the beginning, this means that these lines belong to the field study before. Yes?
How are the columns separated?
No, the columns are actually the characters in this file. So this is a very strict. So if you count the characters in a line, you have either a letter or a word, or you have a blank. So and that means if you need more lines for a field, you just leave out the things in the first 10 colors. That it means you continue the former thing, and then a code in column 6 indicates the type of feature key in an entry in the feature table. So this is a very rich thing to list all type of annotations. This can be there, but don't have to. Okay. So then we have some kind of numbers to describe the thing, and we will see this actually in the yeah. Okay, let's go to I will show you here later on some things. This is only for completeness reason, and it's easier to understand if you then go to the specific suggestions. So these are a few important field types, because if you have to write your parser on your own, you should know what is important and actually what is the meaning of these different types, because sometimes you have to extract the things, and of course you should know where this can be. So type locus, that means it's a short mnemonic name for this entry. This can change actually if the sequence changes. So this has to be present, so that means it's mandatory, and there's only one line. So that means if you find a GeneBank entry file which has 2 lines starting with locus, there is something wrong. So then definition should describe the sequence. This has to be present, but it can has 1 or more lines. So we can have several definitions lines. Then accession. The primary accession number is unique, and an unchanging identifier assigned to each GeneBank record. So this is it's like a personal ID for a sequence, and should never ever change actually. This is also used for citations. Okay, this is mandatory. It can have also 1 or more records. Now you might wonder, okay, how can this be if accession number, okay, suppose is shorter than a line, and how can it be that we need 1 or more records? That the thing is, only the primary accession number is unique. That's typically the first one. But for example, if you have genome project and one large chunk of DNA is assembled by many short ones, so that means the first one is the identifier for the whole thing, and then you just add up all the secondary accession numbers for the short fragments which have been merged into this large one. Okay, and a more useful thing is the version number. So the version number consists of the accession number plus a version. So every time this sequence changes, the version number is updated. So that means it's hard to tell from the file if the annotation or anything else changes, but if the sequence changes, you increment the version number, and typically if you don't specify it, you always get the latest version, but on the version number you can also see changes in the sequence. Okay, yeah. So the primary accession number plus a numeric version. Yeah. So there has been a GI number. It was just maybe it was the abbreviation for gene bank identifier. Was always assigned once the sequence changes. So this could be that different versions of the same sequence had different GIs. I think they removed it now in the March release. So this is no longer supported. This was already deprecated for a long time to use this, but I think if you work with sequences now, you will still encounter this a lot. So this was a kind of old, or is an old identifier which is still present many times, but they just enumerated the sequences, how they came in, and yeah. So version is also a mandatory keyword, and exactly one record, that means one line with one entry is allowed. Then you have several additional things. For example, this DB link. That means you can link out to other databases, because maybe this is an entry in GeneBank. You want to link to a PubMed entry, or you want to link to a entry in SwissProt, and so on. And this all can be found under the DB link thing. So this is actually, if you do a data-driven preparation of a maybe new prediction tool, and you have to find out, or you first want to collect maybe useful data, so you have to walk through different databases and say, okay, I want to find everything which is associated with maybe renal diseases, or kidney diseases. So that means you have maybe one particular gene, and then you start iterating this. Okay, do you have references to other genes which are maybe also involved in this disease, and so on. And this requires that you maybe also check out different databases, and with this DB link fields you can easily navigate through these different databases. Okay, and we have also a keyword section. These are short phrases describing gene products or other information. Okay, so if it's annotated, it has to be there. So of course you can also maybe submit so far unanalyzed sequences which are not annotated yet, and then this is missing, but actually it's if it's already analyzed data, it should be there. Okay, the source means the common name of the organism, and the organism itself, that's the formal scientific name of the organism in the first name, and taxonomic classification. So this helps you, for example, if you start with maybe with a certain organism, and you want to check other similar organisms, and you cannot remember all the described species in your head, you can go there, and then you can say, okay, let's see, are there other organisms in this genus, or to which family belongs an organism, to which order, and so on. You can always go up to larger taxonomic units, and increase your search radius. So okay, this also mandatory sub-keyword, all annotated NS can have 2 or more lines or records. Okay. Again, it can also be here for the source. You can have one sub-keyword or none one. Okay, references, that's for the citations. Okay, so one important thing which is nearly a data structure in this data entry, it's a so-called feature table. Actually, this text format does not really provide that much structure, and you cannot paste a full publication in this text. So people came up with something called a feature. That's a table, and this tries to describe all types of, or holds all types of known information about this gene. For example, if it's only a section of a gene, which sections code for proteins, which code for RNA, sites of biological significance, maybe hotspots for mutations, crossover, and so on and so on. Then actually one important thing is this origin tag, because this marks the start of the sequence, and this is again a mandatory feature and can have exactly only one record. And then of course the sequence data follows without any special field identifier anymore, and of course for the sequence we can have multiple lines, because it can be more than 80 characters, and every sequence entry ends with this double slash. That's the termination symbol, and is also mandatory and can help you to separate these sequence entries. Okay, so this is the thing in how is this specified actually only for this locus line. This locus line is the first line in gene bank entry. So you see columns, actually this is equivalent to characters. So you see in the locus are exactly 5 characters, so that's always, the line always starts as locus. Then you have 6 blanks, 6 spaces, then the locus name, again 1 blank, then the length of the sequence, space, and here a few allowed keywords, and so on and so on and so on. And depending on the specific line type or field type, this can be quite complicated. This is just to give you an idea. So if you really have to write a parser, you should go to the specification and go to the details. Fortunately, I think there are already a few parsers out there where people have already implemented most of this. Okay, last word to the accession numbers. You can have two ways. Old ones have 6 characters or 8 characters, so that means it starts with a single uppercase letter and then 5 digits, or 2 uppercase letters and 6 digits. It's simply because at some point they realized, okay, we are running out of numbers, we have to increase it. Okay, so again, remember, the primary accession number is always the first one. Okay. Okay, that's here. That's a link to this feature table. What you can annotate and how do you do it, and it allows also hierarchical relations between the different features. Again, here you can also include cross references to other data collections. Okay, so it has its own layout rules. So you have a header called feature, and then location qualifiers, and then each line describes a feature key, has a feature key and the location, and can have also multiple things, but that's already quite complicated structure, and if you can, you better use a parser. Okay, so for example, you can have here the decoding region for amino acids in a certain thing, including the stop codon. You have exon boundaries, or a gene if you want to know something about functional gene name, including upstream regulatory things or downstream control elements. Okay, so currently we have more than 60 features. Yeah. Okay. Yeah, for example, location is already a very complicated one, and it can be a single base, it can be a span, a stretch of bases, it can be between between two bases, and it can be a join of sequences. That means sequence 23 to 56, 87 to 110, and so on. So that's, I think that's the attempt to have a rich structure in this, but of course it makes it complicated to have it in text form. Okay, yeah. Qualifiers and yeah. Okay, this is here for the cross-references. That's something you should pay more attention. So actually, because that's always the same thing, you have here the database reference, then the database name, and then the identifier. And if you parse out this, it's very easy to take this and then immediately search the reference database to get more information about this. So for example, this could be then reference to a Swissprot entry. You see here that's the syntax dbref, the name is Swissprot, and that's the Swissprot accession numbers. So especially if you want to cross over between different databases, here, yeah. The only thing is the list of available databases sometimes changes, and yeah, once in a while maybe you should update your infrastructure or your scripts to make sure that you're still, your database are still available, or they did not change, or maybe some new came up. So oops, okay. I think I will increase this. Can I increase this? Let's see. Wait for a second. So here you can see an example of this. So for example here, this is an example for this locus line. Every GeneBank entry starts with a locus line. So you see here also what this means with this yeah, fixed format. So that means I think it's column 1 to 10 is always used for field descriptor. So this is locus, then you have spaces, and then here this other very, very condensed information definition. That's the accession numbers. You see here that's an old style accession number with only one uppercase letter and then 5 digits. Then the version number added with this .0, that means version 1 of the sequence. And here that's the old GI number. We don't have any keywords. We have the organism as a sub keyword from the source, and this is here this taxonomic yeah, hierarchy. So starting with eukaryota and then ending up with the genus Saccharomyces. Okay. Reference means which bases actually we have, author's title, and so on. That means this is referenced by these authors. The title of this publication is this year, where is it published, and so on. And the reference things can be more than one. So there is a second publication about this from different authors. They found here another protein here. Okay, so then let's have a look here at this feature table. Actually, the feature table is introduced by this features label on the left side. Here, the right side starts with the location qualifier pairs. Again, where it's from, so there is some kind of redundancy. But I think this also reflects several evolution steps of the file format. So you have to say the organisms. Here, that's the reference to the taxonomic database. So that's the ID in this taxonomic database. Chromosome, start, codon 3. What is the gene product? And then you have also here the accession numbers for the protein and the putative protein sequences. Okay, and again, for example, you have here another, here it's like a back reference also to a thing in GeneBank with a GI number. Here for this Here, that's another for the GI number. So these are, I think, shorter stretches out of this large contig. Okay. So let's continue here. So this was a little bit more in detail. Again, here the locus line, accession number, GN number, feature tables and annotation. Yeah. Now I want to, besides the file format, I want to cover a little bit other useful resources from the NCBI. How many do I have left? Okay. Yeah. Okay, so first of all, it provides a very good electronic bookshelf, so you can access this via the fact sheet and book and NCBI manuals. Sometimes they have textbooks, might be recent, might be a little bit outdated, but at least they describe the services very well from the NCBI. So they offer certain processes. Why do you need processes? Actually, because that's a database which should be used by all or by a lot of different researchers. It's necessary to standardize some processes. And for example, they offer a prokaryotic genome annotation pipeline which helps you to execute all the necessary steps in the same order. And if the people working in this field use all the same process, this ensures that you can compare the results, and this helps to ensure certain quality. So this was designed to annotate and analyze bacterial and archaeal genomes, and it actually includes a multi-level process like looking for protein coding things, ribosomal RNAs, tRNAs, small RNAs, pseudogenes controls, and so on, and already provides also the tools to predict ab initio functions and proteins based mostly on homology. Okay, another very useful resource is RefSec. The thing is, this is a comprehensive, integrated, non-redundant, and well-annotated set of sequences, including genomic DNA, transcript, and proteome. So, first of all, or let it the other way around. For bacteria, there is a system where you can only describe a new bacterial species if you deposit a type strain. So that means as a type strain, you send it to a collection, and they put it in the liquid nitrogen freezer, and whenever somebody else want to analyze this and want to compare a new isolate with this, you can take out a sample and propagate it, and since bacteria multiply by clonal propagation, so that means they are all identical on the genetic level, you can really compare to this type strain, and you always should get the same genetic information. With very diverse, or with species like humans or plants which have more than one allele and even sexual reproduction, it's very hard to define a type specimen or a type sequence, and so the RefSec has the task to consolidate all this different sequence information and say, okay, this is our reference standard. Because if you relate, or if you want to publish something, you have to relate things. First of all, you have simple question like, is this a wild type sequence or is this a mutated sequence? So you have to first figure out what is actually the wild type or the true sequence. Are there maybe more than one wild types available? Yeah, and working with DNA, you have to give positions. So, and if you ever worked with DNA sequences and you, people start at different points, it's very difficult. So that means, and this provides a consolidated sequence with a consolidate numbering scheme. So they number maybe chromosome 1 to 2 million, and if you relate to a position here in the reference sequence, you can always guarantee that other people can reproduce your work. So that means that's like that's a common standard. Okay. Yeah. So, and this is also accessible via BLAST, Entrée, FTP things. Yeah. So how do you come to this RefSeq? Actually, there is not only a prokaryotic genomic annotation pipeline, but also a eukaryotic genome annotation pipeline. So eukaryotic genomes are a little bit more complicated because you have introns and exons, and you have to handle this allele thing, and you have manual curation. So that's one of the real points where you add benefit. But this also limits the growth and the size because manpower is expensive and it simply takes time. We will see a curation process in detail when we go to SwissProt next week. And yeah, but it means when you take this, you can be very sure that an expert had a look at it, and it's not if you simply download sequences, could be out of a sequencer. Which did not work correctly at this day, so you had a lot of noise in the data, but in the RefSeq you could be quite sure that you can rely on this data. Okay. It's easy to identify RefSeq sequences because they contain an underscore in their accession number. Okay, so here is a short overview about the growth. So this is a different species. You have a RefSeq for proteins, total accession numbers, nucleotides, and you see already that the growth and the number here is still much lower than the ones of the gene bank growth itself, because curation takes time and needs care. Okay. So I think that's a good point to make a short break of 10 minutes. Yeah, let's be back here 5 minutes to 11, and then we'll continue with the other features of the NCBI website. Thank you.